So today, the two lectures today are going to start with a review of the cellular neuroanatomy that we worked on last week, and then a real quick review of what happens during action potentials so that we can get into the propagation, how action potentials move like a wave along an axon from your fingertips to your spinal cord, or from your spinal cord to your toe muscles. And then we'll talk about neurotransmitter release mechanisms. Then by the end of the day, by the end of the second half of today's class, we'll have introduced some neurotransmitters and some important neuroanatomy. Those of you who saw the brain come out of the skull yesterday have seen the brain, and now we're going to talk about its important structures and functions. Well, not now, but soon. So here is a cartoon of two neurons. Dendrites are where the receptors mostly are. Dendrites and soma. The axon hillock is where that input information is integrated and decides whether to fire an action potential, and if so, how many. So input, integration, output of action potentials. The propagation of action potentials is what we're going to be talking about in just a few seconds. These cells in the peripheral nervous system are Schwann cells. They wrap the axon with myelin. So the name of the cell is Schwann cell. The name of the material that wraps the axon is myelin. And you'll see that myelin, or myelin insulation, is important for speeding action potential propagation. There we have neurons, we have axons in our nervous system that are unmyelinated. Some are not myelinated. Most of them have myelin because we have evolved this mechanism to speed action potential propagation. But some are unmyelinated, and we'll talk about those first because it's simpler. Action potentials propagate into synaptic terminals where neurotransmitter is going to be released and then interact with the receptors on the receiving cell, which will input this information and decide whether or not to fire an action potential, etc. So that's a real quick review of the cellular neuroanatomy. Now we're going to focus on the axon and its mechanisms. So at the axon hillock, input, so this is a experimental version of a synaptic input to this cell. This is experimental, but an EPSP, is never square like this and it's never this long, but an EPSP is a small depolarization, and if the small depolarization is big enough, it will start to fire an action potential, one or a train, a series of them. So a depolarization causes a depolarization of the neuron. This one reached threshold. Threshold is the voltage at which enough voltage-gated sodium channels have opened, enough to cause the rapid feedback, positive feedback loop. Sodium channels open, sodium influx causing depolarization, depolarization opening more sodium channels until they are all open and we get to the peak of the action potential. Then the voltage-gated sodium channels inactivate and the voltage-gated potassium channels activate and accelerate the repolarization. And depending on the kinetics and the numbers of channels, we might get after hyperpolarization or not. Depending on the kinetics and the channel expression, we might get just one action potential or a few. Depending on the stimulus, we might get slow or fast trains of action potentials. So all of this is controlled by the number of channels and the specific kinetics of the channels in that cell. But the basics are the same. Voltage-gated sodium channels responsible for the upstroke because of that positive feedback loop that is then broken. The loop is broken by the inactivation of channels and the activation of potassium channels to bring the membrane potential back down. And then, in this case, the cell depolarized again to threshold. Now you might notice, some of you sharp-eyed students will notice, wait a second, there is an action, you said action potentials are all or none, and yet there's one that's different. We're going to talk about that difference in a few minutes. For the most part, they are all or none. It depends on the number of available sodium channels that determines the peak. So each cell has its all, but not all cells have the same all because they have a different number of voltage-gated sodium channels. So let's talk now about how action potentials, once triggered at the axon hillock, propagate along an axon, and then get to a synaptic terminal. That's where we're going now, is propagation of action potentials. So here is a highly idealized cartoon of the membrane of an axon. Membrane, voltage-gated sodium channels. This is one membrane. So remember, this is a tube, and we're just looking at one membrane of it. I mean, it's a tube, and we're looking at one piece of it. So the tube is running this way, and this is one membrane. So this is the axon hillock is over there, and the presynaptic terminals that it's going to is over here. And we're now going to talk about how an action potential gets from here to here. So the depolarization that happened upstream, either because of a EPSP or because action potentials happened over there. Anyway, some depolarization happened and opened voltage-gated sodium channels in this section of the axon. Sodium flows in, that causes further depolarization. That charge moving from the extracellular to the intracellular compartment. Remember, this is the axoplasm, the cytoplasm of the axon here, and this is the extracellular place. This influx, this depolarization, this charge diffuses. And this positive charge, it's sodium, diffuses, and it neutralizes the negative charges on the inside of the next section of the axon. Right? Positive charge neutralizing the resting membrane potential here. Remember, this part of the cell is at rest, and this part of the cell has just fired an action potential. This action potential depolarizes the nearby areas. So what's going to happen? Well, the next slide shows that charge diffuses and charge causes depolarization of the axon. So this charge diffuses, neutralizes the negative charge around this patch or region of sodium channel, and it opens. So we have just allowed this influx to open this channel. This influx, diffusion of charge, depolarization, opening up the channel. So the action potential has moved from here to here. This influx, charge, diffusion, this one diffused both ways too, but we ignored it. Now we're not going to ignore it. This charge, influx, diffusion. This charge diffusion happens, randomly. It goes everywhere. This charge diffusion in this direction, you can see, is going to depolarize this patch, this area of membrane, and it's going to cause this channel to open in the next image. But you could be saying, well, so why doesn't this diffusion open this channel and cause the action potential to go backwards? And the answer is, remember that sodium channels, after they activate, they inactivate. And so this channel is refractory. It's resistant to opening because it is in the inactivated state. It has not yet had enough repolarization, and it has not yet had enough time to recover from inactivation. So this channel is inactivated, and the inactivation of the channel prevents the action potential from propagating in that direction. Okay? Influx, diffusion, inactivated channel, closed channel. This channel depolarizes. And we're going to move to this one next. But I used a word that I want to unpack a moment. I said this channel is refractory. So there are refractory periods. There are times called refractory periods during which action potentials are inhibited. So here is an action potential in time, right? In time. This is an action potential voltage as a function of time. During the action potential itself, pretty much the duration of the action potential itself is called the absolute refractory period. The cell is absolutely refractory. Why is it absolutely? So what does that mean? It means that if we were to give this cell a huge depolarization anywhere in this interval, it still would not fire an action potential. It's refractory to firing an action potential. That means no matter how big a stimulus we give it here, it's not going to that stimulus is not going to trigger another action potential. Why? Because all of the sodium channels have already been engaged in this action potential. They're either open, they're either opening, or they're open, or they're inactivated. So during the absolute refractory period, no matter how big a stimulus you give it, it will never fire another action potential. It's already firing an action potential. During the relative refractory period, which follows the absolute refractory period, the cell can fire an action potential if you give it a bigger stimulus. Why? Because some of the voltage-gated sodium channels have recovered from inactivation. Some of them. More and more and more and more as we move along the relative refractory period. So the after hyperpolarization, if there is one, or the period after an action potential, even if there isn't an after hyperpolarization, is relatively refractory. The cell would need a bigger stimulus in order to get an action potential going again. And during the relative refractory period, because only some of the sodium channels have recovered from inactivation, what does that tell you about the amplitude of action potentials during the relative refractory period? They're going to be smaller. So we see that in this experiment. This action potential happened because this depolarization was big enough, but this cell is still in its relative refractory period at this point in time after this action potential. And that's why this action potential is shorter than this one. Some of the voltage-gated sodium channels are still inactivated. The depolarization was big enough to trigger it, but its all or none is smaller than this all or none because some of the voltage-gated sodium channels are still inactivated. Yeah.
The time between the second action potential, the refractory period, is it shorter than the ones in the end because it went twice?
I'm sorry, could you say that again?
The gap between the spikes is
Mhm.
shorter in the first one than it is in the last one. Is that because it spiked twice?
The gap between this one and this one,
No. The second one and
the gap between this one and this one?
yes.
Um,
Does that have to do with anything or?
so the gap between this one and this one is shorter than the gap between this one and this one. Because, no, that's a good question. I'd have to think about that and try to explain that from a channel perspective. But it's clear that as you depolarize more, you move action potentials closer together. And so this interval of time is more depolarized than this interval of time, and that's probably why. So, after the relative refractory period, during the relative refractory period, if we were to give the cell a big stimulus, it would, so if the membrane potential is going from like this, it would depolarize and it might get to threshold and it might fire an action potential, but it's going to be, you need a bigger stimulus to do this than here, and this action potential is likely to be shorter than this one. And then if we stimulated it here, we would get an action potential that might be closer to the full size because more voltage-gated sodium channels have recovered during this interval of time. And if we give it a normal stimulus here, it's going to give us a normal action potential. Once we move out of the relative refractory period, the cell here is just like the cell here. So action potentials have spread from here to here to here, from here to here to here, and they do not spread backwards because the channels behind the action potential are refractory, are in the refractory period. And so action potentials typically propagate in one direction, from axon hillock to synaptic terminal. But I could ask you a trick question. I could put up this image, and I could say, what if this cell got an artificial stimulation right here? At this point in time, let's pretend that this channel doesn't exist. All the channels are like this. The cell is at rest. And if I were to artificially stimulate it here, which direction is the action potential going to propagate? To the left, to the right, to the left, or both? Stimulate a resting axon here in the middle. Anybody want to shout out an answer? Both. It's going to move in both directions because the cells on both sides of it are ready to go. And so there is such a thing as retrograde action potential propagation, but it's unusual. The usual flow is from hillock to synaptic terminal. from axon hillock to synaptic terminal. So this is another cartoon that just shows the action potential moving from place to place. The green zone is where voltage-gated sodium channels are open. The white zone is where voltage-gated sodium channels are closed. And the red zone is where voltage-gated sodium channels are inactivated, preventing retrograde propagation. In an unmyelinated axon, which you have, the typical speed is about 1 meter per second. That's a typical speed of an unmyelinated propagation. So, let's pretend my arm is 1 meter. It takes a second for an action potential to propagate from here to my spinal cord, and maybe another fraction of a second for it to get to my brain. That's pretty slow. So we have evolved mechanisms to speed it up. We, being the animal kingdom, have evolved mechanisms to speed it up. The two mechanisms that the animal kingdom, and I'm talking about all animals here, the two mechanisms that all animals use to speed conduction are either to increase the diameter of the axon, because large diameter axons conduct faster than small diameter axons. I could get into the biophysics of it, but I don't. Even if I were teaching a whole semester, I don't. It's complicated biophysics. It's a simple equation, but it's biophysics, and we're not going to get into it. But it's true, biophysically, that large diameter axons propagate faster. So animals like mollusks, squids, octopuses, they don't myelinate. If they need to propagate fast, they make big diameter axons. We, animals, mammals and other animals, evolved a mechanism for myelination. We evolved Schwann cells and oligodendrocytes to insulate the axon and speed action potential propagation without getting the axon real big. But still, even in our bodies, the fastest nerves are large diameter and myelinated, and the slowest nerves are small diameter and unmyelinated. So you've had the experience. You're nailing something into a piece of wood, you hit your hand, you feel it. You know you have hit your hand. It doesn't hurt yet, but it's gonna hurt. You know it's gonna hurt. You have about a second before the pain is sensed, because the pain nerves, the nerves in your hand and your arm that carry pain, are typically slow. They're unmyelinated. So you hit your hand, and the fast information, the touch, pressure, hit information gets to your brain, and your brain says, okay, that's going to hurt, and a second later, the pain gets there. Because fast nerves got the information to your brain about being hit, and the slow nerves brought the pain. So let's talk about myelination a little bit more. So here's a cartoon of an axon, a tube. Here are the nodes of Ranvier. Node of Ranvier, where the axon is not covered with myelin. So here is the node, and here is the myelinated region. In this cartoon, this node has just fired an action potential. Sodium is flowing in through the voltage-gated sodium channel. It is depolarized. It is in the midst of this. That influx of sodium and the diffusion of charge is improved because of the insulation in the myelin sheath. Because this axon is insulated, this charge can diffuse further before leaking out. And so this signal, this depolarization here, can be detected by a node of Ranvier a full millimeter away. So that's faster. So if we were to do it unmyelinated, we would activate this patch, and then this patch, and then this patch, and then this patch, and then this patch. Let's just say it like that. There would be 1, 2, 3, 4, 5 patches in an unmyelinated axon. And the diffusion and then opening of voltage-gated sodium channels and more sodium influx, and then diffusion, and then voltage-gated sodium channels, and then influx, and then diffusion. That process is slow. It turns out that diffusion of charge is fast relative to the conformational changes needed to open voltage-gated sodium channels. They're slow. Diffusion of charge is fast because one positive ion repels another positive ion and sort of pushes. There's sort of this daisy chain of positive charge repulsion that accelerates the movement of the charge. If it's unmyelinated, the charge leaks out through the membrane. But the myelination, insulation, allows that charge, that diffusion, that flow of charge to propagate fast and efficiently. So there's an interesting, the concept here is illustrated in this movie. Let me go back. I'm setting this up. So here is an unmyelinated axon, and each of the dominoes represents voltage-gated sodium channels. So an action potential here would cause the domino to fall, and that would trigger an action potential here, which causes the domino to fall, etc. You know what happens here. Here is a myelinated axon. The myelin is represented by these rulers that connect this domino to a domino here. Because this domino is now more efficiently connected by myelin-assisted diffusion, you can already predict what's going to happen. The myelinated axon propagated the action potential much faster. Yeah.
How many action potentials does it skip?
Oh. The question is, could we make this myelin longer and increase the speed even more? And the answer is yes, we could, but the risk is the longer this stretch of myelin is, the greater the risk that an action potential here is not going to get all the way to the other end. And so we have evolved sort of a happy medium between speeding it up enough to do what we need to do, and not making the myelin too long where there's a risk of propagation failure. Did that answer your question? Say again?
How long is the typical myelin section?
Oh, from 0.25 to 1 millimeter in length. The question was how long is the myelinated region. And it's less than a millimeter, half a millimeter typically. The node of Ranvier, so this cartoon is screwed up in the sense that it makes the node of Ranvier look big relative to the myelin. Here's the way it really looks. The node of Ranvier is very small relative to the length of myelin. So the node of Ranvier is much less than a millimeter. You know, I don't know. A few micrometers in size compared to hundreds of micrometers in length. So this is the node of Ranvier. Here's the axon. Axon gets a little bigger actually underneath the myelin. Node of Ranvier, where the voltage-gated sodium channels are. This gray haze are the layers of myelin. The voltage-gated potassium channels are near the voltage-gated sodium channels, but they're not all mixed up in most myelinated axons. They're close enough. These voltage-gated potassium channels are close enough to cause the repolarization here when they are open. Okay. Actually, so I want to remind you that a nerve that we see on the body, a big nerve or even a small nerve, is not one axon. It's a bundle of hundreds or thousands of axons, some of which are big and some of which are small, some of which have thick myelin and some of which have relatively thin myelin. And the size and the thickness of the myelin factor into the speed of conduction of a nerve cell in this nerve bundle. Don't confuse a nerve that we see with one axon. We cannot see one axon with our eye. Even one of those small nerves that we saw in the cadaver is a bundle of hundreds of axons. Okay, so I want to stop talking for a minute and I want to give you an assignment. We're not going to break out into breakout. We're going to do that here. I want you to try to remember, either from your reading of the assignment for today or from self-is, the steps in the release of neurotransmitter. And this cartoon gives you some guidance. So the first step is here, illustrated by this arrow. And the second step is here, related to these channels. And the third step is here, related to these channels, which are actually the same. And the fourth step, and the fifth step, and then the sixth step. We'll take a break here from me talking. You can walk around, you can talk to each other as long as you maintain safe distance, and see if you can write out the sequence of events.